Imagine looking at a beautiful lake or a breathtaking sunset. It's wonderful, right? Now picture a blur spot growing bigger and darker in the center of this picture. This is how your vision would be affected by Stargardt's disease.
Stargardt disease is an inherited degenerative condition of the retina, which leads to macular degeneration and eventually to blindness.
Stargardt's disease is caused by loss of function mutations in the gene ABCA4, which usually eliminates toxic byproducts that may damage the retina.
To date, Stargardt disease is considered an incurable condition, but things may change quickly.
In a recent study published in Nature Medicine, a team of researchers led by the University of Basel and in collaboration with Beam Therapeutics developed a promising approach to treat this condition.
The researchers observed that most Stargardt patients present a G2A mutation in BC4. This is, as you know by now, an ideal target for base editing.
Now, we won't go into details on how base editing works, as we discussed it already in our Making Cut series. So if you want a reminder, go and check it out. Are you back? Good.
In this paper, the authors initially tested multiple guide RNAs for adenine base editing in vitro and selected the best one, and they could achieve up to 40% restoration of the wild type sequence.
The next step was to deliver the base editing using an AAV vector, the most used tool for retinal delivery, even in clinical trials.
As you probably remember, AAVs have limited packaging capacity, and the base editor complex would be too big to deliver in a single AAV.
To solve this issue, the researchers turned to inteins. These are Velcro-style protein segments that allow two protein pieces to be fused together. And using inteins, the scientists managed to split the base editor in two parts that can reconstitute inside the retinal cells.
This way, they could package the base editor complex in two separate AAVs and could show in different cell models, including retinal explants, the same editing efficiency, if not higher, as in vitro.
As the last step, they optimized the AAV vector to efficiently deliver the base editor to the retinal pigmented epithelium, the target cells. Now they were ready to move to an animal model.
In a mouse model, they managed to correct the ABCA4 gene between 20 to 40% in the retina, which resulted in up to 80% of the wild-type ABCA4 mRNA.
And they didn't stop there. They also tested their approach in non-human primates, which are the closest possible model to a human eye. And in these animals, they could show up to 40% correction of the ABCA4 mRNA.
Last but not least, they ran an extensive target profile to ensure that no unwanted and potentially harmful modifications were inserted by the base editor in the process of correcting the Stargardt mutation.
The results they obtained are quite amazing, and they bring us closer to a potential treatment for a condition that so far had none. We here at With CRISPR will follow this topic closely and be ready to tell you all about it.